In chapter 20, we're continuing to learn a little bit more about plants, looking this time mainly at their structures and how they are able to transport water. So the organization of a vascular plant, and again in chapter 19, we talked about how through the evolution there were non-vascular. So we are focusing just on vascular plants here that have a vascular tissue. We'll talk more about xylem and phloem coming up. So these vascular plants have are organized on a vertical axis. So everything below the ground is the root. I think we know this by this point. Of course, that root penetrates the soil. Its main job is to absorb water and ions. And then its other main function is to anchor the plant. So absorbing nutrients and water and anchoring the plant. Then everything above ground is referred to as the shoot, and this would include the stems and the leaves. The stem is more the framework, kind of provides that structure and support. And then the main job of the leaves is to perform the photosynthesis so that it has food and energy to support its growth. So the body of the plant, again, everything above the plant here is the shoot system, and then everything below would be the root system. Within the shoot system, a few different things just to go ahead and talk about here. Of course, how it grows is called the apical meristem. So this is going to be primary growth. We'll talk about it. So there's a meristem here in what's called the terminal bud at the very top of the plant. There's also an apical meristem at the very bottom of the root that allows the root to continue growing longwise. Then if we look at our leaves, the leaf is made up of the blade, and then the veins, of course, have that transport system. Where it attaches is called the petiole. Where new leaves grow from is called the axillary bud. And then we also have what are referred to as lateral meristems that are growing. That's going to allow the cell or the stem to continue growing laterally. A node, again, kind of that point of attachment of the leaf to the stem. And then the internode is the space between two leaves. So like right in here, there would also be another one here. So the space between two nodes, or the internode. And then down in the roots, of course, we have monocots and dicots. So regardless, we have a primary root that has that apical meristem, but then there are also lateral roots that come off. All right, now moving into these plant parts, of course, after the labeling. So we have what are referred to as meristems, and these are growth zones that are involved. So they're not only areas of actively dividing cells, but also continuously replenishing themselves. So they are growing, but also replacing dead cells. So in this way, those stem cells function a lot like stem cells in animals. They can give rise to any type of cell. So they are consistently dividing, replenishing themselves, and producing new cells. So there are different types of growth. So primary and secondary growth. I mentioned the apical meristems. Again, these occur at the tips of our root and the shoot system, so at the very top. And this is going to allow extension of the plant body, so vertical growth, so getting the plant getting taller, the roots getting longer. Then the secondary growth, that is going to be the lateral meristems, and it's going to, as the name implies, allow the plant to thicken, so the stem to get wider. Think about like trees adding on a new annual ring. That's the lateral growth, referred to as secondary growth. So those lateral meristems, and we have actually two different types of them. We just have one for the primary, that apical meristem, but we have two lateral. Vascular cambium. This gives rise to thick accumulations of what's referred to as secondary xylem and phloem. That's the vascular tissue. And then we also have cork cambium, and this is going to be the outer layers of bark that's going to be on both the roots and the shoots. So the most outer layer is the cork cambium, and then think about vascular, which is our xylem and phloem. That's referred to as the vascular cambium. All right, so the different types of plant tissues, I've already alluded a little bit to the vascular, but there are three types. So there's ground tissue. That's where the vascular tissue is embedded. So it's not, doesn't make up the vascular tissue, but it's where it's found. And then we have dermal tissue, thinking about our epidermis that we talked about with animals. This is the outer protective covering. And then that vascular tissue, again, just like in animals, delivering nutrients, kind of the delivery system. So that vascular tissue conducts the water and minerals up the plant, and also the products of photosynthesis like glucose throughout. So that delivery system. Now in the ground tissue, starting with that, we have three kinds of cells that make up ground tissue. We have parenchyma. This is alive at maturity, and it carries out the basic functions. So things like photosynthesis, going through cellular respiration, and then storing water and food. Then the collenchyma, which is also alive at maturity. This is providing support for the plant in which that secondary growth has not yet occurred. So before it gets kind of stabilized with that lateral growth, the collenchyma kind of helps support those plant organs. And then finally, the sclerenchyma. This is actually not living, so this would be kind of dead tissue. And these are essentially secondary cell walls. So this is going to be much more rigid. The cell wall, again, very rigid. But after the cells, again, not really actively conducting, not going through photosynthesis, they're not really containing living parts or active cells. They are now referred to as sclerenchyma. And this is just a picture here showing them, again, parenchyma, thinking about photosynthesis, kind of most of the activity of the plant. Again, the collenchyma, this almost kind of reminds us a little bit of cartilage, but the collenchyma is more that support system. And then the sclerenchyma is mainly just kind of like empty cell walls. There's not really anything inside happening. Again, no photosynthesis, no chloroplast actively doing anything. So again, more of our dead cells within that tissue. Just a little bit more about the sclerenchyma. There are two types of that. There are fibers, which are long, slender cells. And then there are sclerids, and these have a little different variable shape. They're not always the same, but usually they're kind of a branch-like network, and clusters of those give that kind of gritty texture to the flesh of pears. So if you like pears, or at least familiar with them, that kind of gritty texture, that's made up of a bunch of these sclerids. All right, so then our dermal tissue, again, this is the outer covering. So that flattened epidermal, this is the most abundant on the outer layer. So again, that epidermis, same as what we had when we talked about animal cells. Now, it's often covered with a waxy layer referred to as the cuticle. And those both, the combination of the cuticle and the epidermis, protect the plant and also provided a barrier to prevent water loss, especially during warm or hot seasons. Then those epidermal cells have specialized cells. The first one we'll talk about, guard cells. So it implies it's guarding something. Well, it's actually guarding the opening that are referred to as stoma. So in order for plants to go through photosynthesis, they need carbon dioxide to come in, and then they release oxygen as a byproduct. But those gases can't get past the cuticle, so there are small openings called stoma. What allow them to open and close are these guard cells. So they are regulating the passage of those gases, but also water. The trichomes, these are outgrowths from the epidermis. They kind of occur on the shoot, again, above ground, kind of have like a fuzzy appearance to them. And their main job is for insulation, and then that can affect the heat and the water balance. And then finally, the last type are root hairs, obviously in the root system. So this is going to be below ground, but they're going to keep the root in intimate contact with the soil. So it just improves the connectivity. So they play a very important role in absorption. Also increases the surface area, allows the root, because it's going out, kind of like the dendrites of our neurons that spread out to allow more signals to be obtained or received. The root hairs spread out to absorb more nutrients. Just kind of looking at these again here, showing these trichomes. these little projections that stick off more the shoot system that we can see here. Again, kind of a fuzzy appearance. Again, these stomata, so these are the guard cells going around, and it gives rise to these openings for that gas exchange, but also water. And then, of course, the root hairs that we can see growing off of the root. All right, now the vascular tissue, I mentioned a couple times, two types of vascular tissue, xylem and phloem, and mainly their difference is what they're doing. So the xylem is a water conducting tissue, whereas phloem is food conducting. So if we think about phloem starting with the sound F, of course starts with a PH, but we think about the sound F, so phloem for food, or phloem, F for phloem and F for food, to kind of help you remember that. And then the xylem is more about the water and the minerals. So it forms this continuous system that goes throughout, and that water passes from the roots to the shoot. And then whenever it reaches the leaves, most of it actually exits out through those stomata. So it doesn't actually kind of circulate back through exactly the same way we think about our blood. So a lot of that water that gets transported by the xylem is lost out of those stomata. A little bit more about the xylem. So there are two principal types of xylem. There are the tracheids and then the vessel elements. And the tracheids are elongated cells. They overlap kind of at the end. And water flows from one to the other through openings called pits. Then the vessel elements also elongated, but they line up end to end instead of kind of overlapping. And then the end walls are almost completely open, or maybe just a little perforated, and that allows water to flow from one to the other. So the vessels here that have these blunt kind of end-to-end connections, they're much more efficient at conducting that water than what the tracheids are. And this is just showing what these look like. So again, the tracheids have this overlapping region with these pits, these kind of openings here of pits, whereas with our vessels, that vessel element, they line up end to end and allow kind of that continuation or continuous movement of the water through either the pores or just across the membranes. All right, now looking a little bit more at the phloem. Again, this is the food conduction that's carried out. So there are two types of elongated cells here. So sieve cells, these make smaller perforations between the cells or have those. And then the sieve tube members, and these have some sieve areas with larger pores. So again, smaller perforations here in the sieve cells. The sieve tube members have larger pores than the sieve cells. And those are referred to as sieve plates. And those tube members, they line up end to end like the vessels we just talked about to form those sieve tubes. And then the specialized parenchyma, again, the ones actively functioning, they're known as companion cells. They're actually associated with those sieve tubes that are going to dump off the glucose and the carbs that are being made through photosynthesis, and the parenchyma cells, they dump that off to those sieve tubes to allow it to be conducted throughout the cell or throughout the plant. All right, so just looking at this, again, we have the sieve tube member that's in here. We have the sieve plates, again, these perforations that are in here that are a little bit larger. And then again, along the edges, we have these parenchyma or companion cells that are dumping their glucose off into those to then be transported by that phloem. All right, now looking a little bit more at their roots. So they have a central column of xylem that's there in the middle, and then it has radiating arms that go off the side. And they're alternating with those arms or strands of phloem. So the xylem and phloem moving through them. And then they're surrounded by a central column that forms a boundary, and that makes up what's called the pericycle. And so these branch or lateral roots are formed from the cells of that pericycle. So that's how they're growing. So like the root hairs or other lateral roots that are branching off, they form out of that central column called the pericycle. So then the root epidermis, so this is that outer layer of the root. So there's a mass of those parenchyma cells. Again, those are the actively functioning cells. They make up the vascular tissue called the cortex. And then the innermost layer that lies just outside that pericycle, where those lateral roots are coming, that's called the endodermis. So epi, outside, endo, inside. And the cells that make up the endodermis are encircled by this thick, waxy band called the Casparian strip. And this strip blocks the movement of water between endodermal cells and instead forces the movement through the membrane into those endodermal cells. All right, if we look at the cross section here of the root, so of course the epidermis is on the outside. Then we have this layer of ground tissue here called the cortex. And then if we look all the way on the inside, we have that vascular tissue. And the ring of vascular tissue that goes around the outside that separates it from the cortex, that's called the endodermis. And then the smaller cells that are in here, those are going to be the primary phloem. These larger cells that we see in here, that's the primary xylem. And then you have a few of these pericycle cells where those lateral roots would start coming from. And then if we just highlight this a little bit, we can also see this Casparian strip where those endodermal cells are, where that water is being pushed or forced through. All right, so now looking at the apical meristems, again, this is what's giving rise to that primary growth, the vertical growth. So we have a couple different types. So we have protoderm, that becomes the epidermis. So if we think kind of about, again, stem cells that can become anything. The protoderm are the meristems that can become epidermis. The procambium, that produces vascular tissue like the xylem and phloem. And then the ground meristem, as the name implies, gives rise to the ground tissue that help, including those parenchyma cells. So if the apical meristem growth is outward, then cell division forms kind of like a thimble mass of unorganized cells that make up a root cap, kind of like a mole almost on us. So if it's kind of growing outward, we get these little root caps, and they protect that apical meristem as it's growing through the soil so that it doesn't get damaged. Because if it gets damaged, if we think about like our growth plates, if that root cap or those meristems get damaged, then it can't get any longer or have any more vertical or vertical growth. All right, there are also what are referred to as root zones. So these roots elongate rapidly right behind the tip. So if you've been in lab where you've looked at like an onion tip looking at mitosis, the reason that's a good place is because right at the tip, right behind that apical meristem is this zone of elongation, because those are the cells actively dividing to make that root longer. There are also abundant root hairs there for, again, absorbing water to support the growth, with the extensions of single epidermal cells. And so that area is referred to as the zone of differentiation. And then, so if we look here at our root structure, comparing the monocots and the dicots. So if you remember, dicots are a taproot. They go down, they have a single big root with lots of lateral roots coming off, whereas the monocots have multiple roots about the same size going down into the ground. So if we look first at the dicot, again, we've kind of already looked at this, but again, we have this root cap here that protects that apical meristem that's right behind it. And that apical meristem is giving rise to new cells. Again, our procambium that's giving rise to that vascular tissue, the ground meristem giving rise to ground tissue, and the protoderm that's making that dermal tissue. Again, we have the xylem and phloem in the middle that make that up. Again, our endodermis that forms a ring around that vascular tissue, the pericycle where new roots would grow from, some of our little root hairs, and then ultimately, again, that xylem, the larger cells and the phloem in green, which are the smaller ones. Again, our ground tissue here is called the cortex, and that outer layer of the epidermis. And then again, our zone of elongation, that's where that apical meristem is going to be. So it's just growing and extending. Whereas behind it, this zone of differentiation, that's where the cells are becoming ground cells, or they're becoming epidermal cells. So again, they're differentiating. They can't become any type of cell. All right, now if we look at some of these lateral roots that come off, our picture here shows a developing lateral root. So again, that pericycle that's just inside of that endodermis, that pericycle gives rise to new roots there, or those lateral roots. So this is showing one of those appearing. So as they start to branch out, but again, it's coming from that pericycle, and that developing root just grows out of the cortex toward the surface of that root. All right, now moving on to our stems. They have both primary and secondary growth that's occurring. So these are the source of economically important products, mainly wood. Of course, not all plants are wood plants, but they definitely have a huge economical importance to us. So the primary growth of a shoot, that first appears as what's called the primordia, that occurs. These are rudimentary leaves, so they're not true mature leaves yet, that are clustered around that apical meristem, and then they unfold and grow as the stem gets a little longer. All right, so then the nodes that we mentioned earlier, this is where the stems are going to give rise to the leaves. So the leaves are attaching and coming out from the stems at what are called nodes. And then the space in between where two leaves are attaching is referred to as the internodes. And as the leaves expand, a bud actually develops at an angle there between the leaf and the stem from where it's coming, and that area is referred to as an axil. Now these buds have their own immature leaves called stipules. So again, thinking about just the development, so the mature leaf. So before they mature, this little immature version is referred to as a stipule. So those buds may either elongate or they may just stay dormant. The leaf may never mature from there. So that activity is controlled by hormones, similar to our maturity. So it moves downward from that terminal bud to the shoot, and that hormone suppresses the expansion of the bud. And then the bud begins forming in the lower portion of the stem where that hormone has been reduced where it hasn't quite gotten to yet. So here just looking at a woody twig, a little bit different than what we'd seen. Our petiole, that's where the branch or the leaf branch is going to attach to that woody stem. Again, we still have our blade on our leaves. We still have nodes where the leaf attaches and internodes. But again, kind of this little leaf branch is new, referred to as a petiole. Still have that terminal bud at the end where that apical meristem is. And then you can also see these terminal bud scars. So at the end of each kind of growing season, so in the fall, whenever it becomes dormant, this apical meristem here falls off and it leaves behind a scar. So kind of like annual rings, you can count those to see how old that little branch would be. All right, so that stem primary growth. So within the soft young stems, that vascular tissue is arranged differently in the dicots and the monocots. So in the dicots, those vascular bundles, again, xylem and phloem, they're arranged around the outside of the stem. And then in monocots, the vascular bundles, they're scattered throughout. So they're randomly kind of dispersed instead of in an organized kind of structure that we see in the dicots. So again, just kind of looking here. So again, this is in the stem. So in the dicots, that xylem and phloem forms these vascular bundles that are formed this nice little ring around the outside, very organized. Whereas in the monocot, those vascular bundles are just kind of randomly dispersed. You see quite a few on the outside, but there's not a true pattern to them like we see in the dicot. Then that secondary growth. So again, this is initiated by the vascular cambium. which is that thin layer of actively dividing cells between the bark and the main stem of that woody plant. So it's right in between the xylem and the phloem. So these cells divide outwardly to become the secondary phloem, and then inwardly, those become the secondary xylem. So out becomes phloem, inward becomes xylem. Then that cork cambium, so while that vascular cambium is kind of being set up, that second lateral cambium develops on the outer part, so on that outer layer there. So that cambium consists of plates of dividing cells. They move deeper and deeper into the stem as they divide. So outwardly, it divides to form densely packed cork cells. Then inwardly, they produce that parenchyma, those parenchyma cells, to carry out their different functions. All right, so just kind of looking at this. So again, we have our cork cambium here, the vascular cambium to give rise to that vascular tissue like the xylem and phloem, whereas again, our cork cambium is there for those other cells, like especially the parenchyma. So we see kind of this green layer indicating those actively functioning cells of the parenchyma. All right, now we have periderm. So that cork, the cork cambium and the parenchyma cells collectively make up a layer called the periderm. So we have the epiderm all the way on the outside, but then the periderm is all that outer tissue. So that's the outer protective covering. Essentially, this is what we call bark. So the true scientific name for bark is periderm as it matures. And then the woody part, that's actually the secondary xylem that was being created. Of course, annual rings we can use to estimate the age of each tree that's out there because of the way it's accumulated. So that vascular cambium that's giving rise to the xylem and phloem, it divides more actively in the spring in the summer than it does in the fall and winter. So because the growth rate is different, it kind of has this alternating thickness, and those different thicknesses provide those annual rings. And of course, we can count those rings to determine how many seasons this tree has been through to estimate the age.